Good morning, everyone. So today we are at our fourth lesson webinar about bioinformatics for our biohackathon. So let me share the screen and let's discuss. Okay. So basically, you already saw a couple of things about reproducibility and containerization with our first webinar, and how to use also the HPC. So whenever it comes, when you will arrive in our venue and you know already that you will use an HPC, you will connect to an HPC in order to perform some analysis. Then you did another webinar about single-cell RNA-seq. So of course, moving the target to single-cell RNA-seq, that means that the biohackathon will be about single-cell RNA-seq. And you learned a few things about wet lab protocol and quality control. So starting from the experiment, how to perform an experiment. Then instead, 2 weeks ago, you start with the computational method, so the part 1 of single-cell RNA-seq. So pre-processing, all the method for normalization and quality control. What we will talk today is we will talk about the focus, the core of the analysis, that is the clusterization. So how to check and inspect our data, how to define which subpopulation we have in our data. So kind of giving a name to each cell that comes from our experiment. And also trying to, and this will be the first part. And then the second part of today, it will be about feature selection. That is, let's check which are the genes that play a major role in these, that are the main protagonist of this separation, of this clustering. The way we organized, I'm Alessandro Luca, and Sofia instead, she will be the next speaker. She will talk about feature selection. Instead, I will talk about clustering. So from this QR code, you can find both of our scripts. So the scripts are actually accessible for everyone, all the scripts that you will see today. In particular, for my part, so both of them, they point to a GitHub. In particular, for my path, you will find that the GitHub is actually pretty full of things. I can show you in a few minutes what it is. So basically, you're going to have a README file that can explain a bit what you will find and how to use it. But in a few words, you're going to have a Docker file. So I believe you already started using a Singularity. Here I'm using Docker. But actually, Docker and Singularity are kind of the same thing. So if you still want to keep using Docker, you can simply use these two scripts to run the Jupyter Lab. We will see Jupyter Lab later, but I already showed you what is a Jupyter Lab in the very first webinar. Instead, for who wants still to use Jupyter Singularity, well, you can run the Jupyter Lab through this script from Singularity. And you have two options. If you have Windows, you can run it with CMD. And if you have Linux or Mac, you can run it through this script. And of course, for Singularity, you can only run with Linux and Mac. Basically, running this script, so you can clone or download this GitHub, and what you will have is exactly the same folder on your computer. So if you have Windows, simply double-click on this one. If you have Linux or Mac, simply just run it through your terminal with /script. If you want to use the Singularity one, /script Singularity. SH. And one thing that you have to keep it in mind is that this one will give me permission denied. So always remember to change the the permission. So give the permission to be executable, the script. I'm not going to run it because I don't have Singularity on my computer, but I mean, this is the idea. Once you run it, you will see a few green things, as you can see from here. something like this. And this is going to tell you that the Jupyter Lab is working and is accessible. How to access a Jupyter Lab? Well, you can access the Jupyter Lab through your browser. So simply open a browser and say localhost 8888. I just want to highlight that this will be one of the two possible scenarios that you will use during the BioHackathon. Indeed, you can use either the Jupyter Lab option or the RStudio option. Just keep in mind that Jupyter Lab, through Jupyter Lab, you can access to both R and Python. Instead, for RStudio, you can access only to R. There is a way, so you can use Reticulate in order to access to also Python in R, but it's not really recommended. So if you want to use Python and R, just use Jupyter Lab. If you want to use just R, also RStudio is fine. In just working on with this environment that I set it up, you can see that here to the left you have a folder that is called shared folder. Well, this folder is the tunnel that connects your Docker, your virtual machine, with your computer. Indeed, if I create a new folder over here, this folder is going to pop out here. So all the data, all the script, Docker file, and everything, they will be present. All the script that I will talk about today, they are in this script. We will see 4 scripts together, and then I will leave the stage to Sophia. And these 4 scripts are about downloading the data, pre-processing data, and then a few words on the 2 clustering algorithm. Basically, yeah, I believe I already gave a good introduction, and we will see other things more in detail later on. So let's talk about clustering in general. So what is a clustering algorithm? Well, a clustering algorithm is an algorithm that divide all of your data, in this case, all of your cell, in groups. How can you describe this? So imagine that you have for each of your cell, and each of this dot is a cell. So imagine that for each of this cell, you have only 2 genes, gene 1 and gene 2. Well, you can represent this really through an Euclidean graph. So simply plotting every cell according to the expression of each of these genes. So let's say that this is 5, then this cell have gene 1 expressed 5, and gene 2 probably will be expressed 1. And here you can represent each of your cell in this way. Well, unfortunately, well, not unfortunately, but like the real raw truth is that human or mouse or other organism doesn't have only 2 genes. They have more than 1 genes. So how can you present in the space? Well, unless you have 3 genes, you can add another axis over here, and then you can still represent it in space. But if you have 4 genes, okay, you can add a color and then represent in 4 different ways. So with the 3 axes and the color one. But what if we have 5 genes, 6 genes? I mean, we have 20,000 genes or even more sometimes. So how can we represent this data through a 2D dimension? Because this is the first question. We're not talking about clustering strictly right now, because clustering is exactly this. So it's deciding how to group a bunch of cells according to their gene expression. So these 3 are 3 different clusters, because the cells are really, they group together all of these 3. But the very first question is, how can we represent a high dimensional space, this is how it's called, in a low dimensional space, that is the 2-dimensional space, so 1 and 2 dimension. How we can do that? Well, there is a very nice example that lets you understand this, and it's the donut example. So if you have a light and you highlight this donut, well, you can see a shadow, and this shadow is basically what? Is the projection of this donut on a 2D environment. So basically you have the donut that is a 3D object, right? Because you can take it to your hand, play with it, eat it if you want. And if you highlight it with the shadow, well, the shadow is shadow on a surface. This surface is a 2D. So, I mean, this is the very first example in how can you project something. Of course, you cannot just take a light and highlight your cell. That would not do the work. I mean, this is just, of course, an example. But just to give you another point from this example, yes, so these two are the component 1 and component 2, as you can see. You can imagine that this is a two-dimensional space. What if we change the donut, or we change the light position? So if we change the lamp position, well, the donut is still the same, but the shape, it will be different. It's still a donut, and it's still kind of similar, but the shape, it is different. Probably from this point of view also, we can see the hole in the middle. Instead, from this one, we couldn't see that. So why am I showing this? Well, I'm showing this because it is really important to define the parameters, and it's really important to understand that whatever clustering algorithm, whatever approach you will use to represent your data, well, they will be correct, okay? Because, I mean, this is correct. This is a projection. This is also correct. This is a projection. They just show you different things. And this is what we will talk about today, okay? Mainly, how to choose a clustering algorithm, which parameter to use, and whatever. So, yes, what is a clustering algorithm? Now you saw how we can project some data. Of course, we will not use a lamp, we will use an algorithm. And this algorithm basically is doing exactly the same thing as this lamp. It's taking some position and is throwing this position over a two-dimensional space. I mean, the explanation is pretty high mathematics, so I'm not going really through this thing, but this is important so you understand why it's called a projection. So once we have this projection, well, we have to clusterize this data, so we have to assign them by groups. Of course, just looking at these three, I mean, it's pretty obvious that these three are three different clusters. But first, we are not a clustering algorithm. We cannot simply just check every dot and say, oh yes, these two dots are very close, so they are a clustering. And we need an algorithm that do that for us. The main problem about this is that there is no ring to rule them all. There is no one clustering algorithm that can perform the same to every data. There is this super nice paper that talks about it. It's from last year, so it's not old. There are a bunch of clustering algorithms that were used, and it's pretty straightforward to even just checking the results to realize that there was no clustering algorithm that was performing as the best on every data that they analyzed. So, I mean, this is a problem, but why there is this thing? I mean, why there is no why this clustering is so hard? I mean, just looking at this, it should be easy, right? Why it is so hard? Well, I can give you another example. So about why clustering is so complicated. Let's say that we have to clusterize these bottles, and then each of you is a clustering algorithm. So I can pick one person from home and ask, okay, can you please divide me these bottles? And then some of you will decide to divide them according to the alcohol percentage. Someone else will tell me, no, this is wrong. You have to clusterize according by size. And someone else is going to say, no, wait. You have to clusterize them according to packaging. Well, I mean, I believe it's kind of obvious. All the three answers are correct. So which clustering algorithm you have to choose? Of course, you have to find the one that better fits your biological question. And this is what is really hard to do, because looking at these three examples, it is pretty easy. If I want to drink alcohol and then I want to decide which between this bottle to drink, I will use this clustering algorithm number one. and I have to decide only to drink either a sambuca or a beer. If I want to drink a lot because I'm thirsty, well, in this case, I will divide them by size, and then I will decide probably to drink either milk or water. If instead I'm really into environmental friendly things, well, then I would like to divide them according to the packaging. And then maybe I will choose the orange juice or the milk, or actually the glass. But you get the idea. The main problem is that we have to choose the clustering algorithm according to the one that better fit our biological question. So already this is a very stressful point, but whenever you will do a single cell RNA-seq experiment, the question has to be clear. So don't do a single cell RNA-seq experiment just because it's cool and because you need a single cell RNA-seq experiment to publish a very well data. If you have to do it, well, do it, but at least think before doing the experiment, because the experimental design is everything from this. Of course, you can always do a single cell RNA-seq experiment and characterize your data, but that doesn't mean that you will always be able to characterize your data. There are other criticalities besides choosing the clustering algorithm. And there is another reason why clustering algorithm is so hard. And we will go through that one in a moment. So thinking about how to choose the best clustering algorithm, it's not like choosing the best statistical test, because, for statistical test, you can choose, you have like some guideline. So you need to have a specific distribution. You need to have a specific amount of data. But for clustering, you are on your own. I mean, if you just type on Google clustering algorithm for single cell RNA-seq, every year there are 10, 20 different clustering approaches, all of them valid. But why should you use one instead of another one? Let's start easy. So there are two main clustering algorithms, two typology. One is the hierarchical and the other one is a not hierarchical. What is the differences? But basically the difference, without going through the math and through the real methodology, in this one, in the hierarchical, it will divide your cell. Let's talk about cell. I mean, we are talking about cell. It will divide your cell through a dendrogram. And then at some point, it will decide, kind of automatically, there are ways, but it will decide where to cut this dendrogram. And so you will have your cluster. For example, we can divide it at this level, and then you will have all of this cell, C, D, E, and F, that belongs to the same cluster, and then A and B to the others. So there are ways, okay? And this dendrogram is a bit more automatic. There is also the not hierarchical. Not hierarchical is more precise from a point of view, and then but have a disadvantage, because with the not hierarchical, you have to say to the algorithm, okay, divide me this data set into 5 different clusters. So you already see the point. I mean, it's really hard to say already a priori, like, which is the best clustering algorithm, like clustering number that I have to use. So now I have more problems. Not only I have to decide which clustering algorithm to use, but I also have to pick the correct number of clusters. And usually, whenever you do single cell RNA-seq, you don't have idea about how many subpopulation you might have, because that's the point of doing single cell RNA-seq, basically. You are exploring a data set, so you don't have a clear idea about how many subpopulation you want to have. So these are the two main problems. Let's go through the code and let's see a bit more about this. So you can find the data set. It is actually the one that used the microleaks from the previous webinar. So you can find the data set from this QR code. And again, this is the QR code for the GitHub in which you will find this script. And this script is the processing data script. First, if you are lazy and you don't want to download all the file by yourself, well, there is a script here that is called download data. And this script basically is going to download all the data for you and will extract them and put it in a folder that is called data. Now it is empty, but I'm not going to rerun all the code again. Of course, it will take some time. But yes, so just expect after to run this code, everything is already installed in this Docker, so no worries about installation or so on. Once you run this code, you will see something like this, and that means that your cells are actually already in order and downloaded in your data folder, shared data folder. Keep in mind that shared data folder in your computer, in your host, it will be in your folder in which you downloaded all the file from GitHub, and then you have a data, and here is the folder. So once the data are downloaded, and this is why you need the first 0 download data. I mean, you can also download them like basically one by one. You don't need to download all of them. Indeed, downloading all of them and clusterize all of them will require a huge amount of RAM. So I'm not really suggest you to run this on your computer. If you want to stress a server and you have a server, you can run it on your server, but don't do it at home. Don't do it on your computer. So this is the first script. Let's go to the second script, because I want to talk about another criticalities that probably my colleagues already told you, but I want to highlight it even more. So you can see that all the files that you download are RDS. So RDS, and this I'm pretty sure you already saw last webinar, is a format in which you can load some table, this count table, right? The main problem is that in order to access to this is through Seura. I'm not really sure if Python have some packages that are able to read this this type of format, but I mean the standard format is the MTX format, and you should. My suggestion is always to stick with that. MTX format is very general. It works with everything. It works with Python, with R, and other programming languages. It works pretty well also if you want to run some code with AI, for example. So just stick with it. Indeed, the very first code, the preprocessing data, is not about saying the same thing that my colleagues told you last time, but is about converting these RDS into MTX TSV files. Okay. So once you downloaded your data, just run this code, and this code really basically just open the folder. I point to the folder in which you have all your data. Take all the files that are RDS, and then of course you have to detect the genes that are common across all the sample. Actually, this step is kind of optional, because if the data that you are downloading are raw data, that means that all the data are generated from the same algorithm, Cell Ranger, for example, the genes will always be the same. So this is pretty optional. But if you have more data set and you have to integrate all of them, I mean, you need to choose either the common one, this is what I did right now, or you have to make another script in order to put a 0 all the genes that are not shared between across all the sample. Okay, so this is pretty important to do. The second one instead is to build a sparse matrix block by block. So basically, you read all the file in your list, you create, you load the sample, and then you just load the exon file. So you will see that this RDS file has not only the exon, also the introns and other important things. So I would say that for RNA-seq, single-cell RNA-seq, just looking at the exon, it is enough. But always keep in mind your biological question. So is my experiment, does my experiment also need introns or other things, or I don't know, whatever. So always, all of these webinars are there in order to make you aware of some criticalities that there are, because code like today is not really such a big deal. Through ChatGPT or Stack Overflow, I mean, you can definitely write this code in a bunch of minutes. but you have to be aware about what you are writing, what you are running, and which are all the criticalities. Okay. So yeah, this very first example is just need this very first code. Just need to take the data and convert into an MTX and TSV and TSV. Well, this is important because RDS, as I said, is a format made from Seura, is a package from Setija Lab. And Seura is, I would say that is the gold standard so far for preprocessing and clustering algorithm, and also some feature selection. It's kind of the standard for single-cell RNA-seq, but it's not the only one. And as we saw before, there is no one clustering algorithm that perform the best compared to like on every type of data. So that's the reason why, even though there is a gold, and most of the people just use Seurat, doesn't mean that it's the best. Okay. So if you want to use other clustering algorithm, well, you have to convert this type of data into an MTX or a dense matrix or something like that. So this is how you use this code. Once you convert this, the table, all of your data in one MTX, TSV, and TSV file, of course, here I'm merging everything, all my sample, as you could see from here. We have several data sets, so I'm merging all the data sets in one MTX and feature and barcode file. And I'm not going to explain again what is this, because I mean, you already saw in the previous webinar. So just take a look. If you have trouble understanding what are these TSV file, MTX, and RDS, please just look the previous webinar, because there it's kind of is explained how to load this type of data. But then there are two other there are two other parts of the code in this GitHub, and I want to say this is wrong. Okay, I just want to show you. But all this clustering, these two clustering algorithms, so these two pictures that you will see over here, also the hierarchical clustering, are wrong, and I'm going to show you why. So the very first thing that I said, let's start easy. So with a hierarchical clustering and not hierarchical clustering. So using the function hclust in R and k-means in R. So of course, for the sake of time, I decided to subsample 500 cell, and of course, always set the seed. What is the seed? So the seed is a value that it is important for computer reproducibility. Whenever I say random for computer, random does not exist. So if I ask my computer to pick a number from 1 to 10, and then I run this function 10 times, of course, and then I ask you to run it 10 times, of course, our results, it would be different. The main thing is that randomness is a concept that computer does not have. So it's using things in order to make it appear random, to have a random distribution, but it is not really random. So this value over here, it is important, because setting the seed, it will mean that you are setting the starting point of your randomness. What does that mean? That means that setting the seed, if I run this code today and I give you this code and you run this code, and in this code there is a random number, okay? So here is like, I don't know actually the random function in R, which one it is, but if I ask R to give me a random number through a random function, maybe it's rand, and then I say, just give me one number. So this value, it will be the same on my computer and on your computer. But if you don't set the seed, well, this value, it will be different. So this is the reason why. And setting the seed is very important for clustering algorithms, because most of the time clustering algorithm works in a way in which they start from a random cell, like this one, start from a random cell, put this cell in a space, then take another cell, evaluate the distance from the two cells, and then say, okay, maybe I can put this one here. And then, so the random, it is very important. It will change the results, not really deeply change the results, but it will change the results. So make sure to always set the seed, or at least make sure to understand if your clustering algorithm requires randomness, and if require randomness and you want to reproduce the same results, please set the seed. Here you can put whatever number you want. Okay? It's not a problem. So here I subsample 500 cells, and then I also detect the 1,000 most variable genes. Just because I believe that, I mean, this is of course another thing that I did that is not for sure correct, but I mean, selecting 1,000 genes, the one that are most variable genes will just, I would think that it will remove the noise, for example, okay? But maybe 1,000 is too less, maybe I want more. So always whenever you do clustering, start big. And then if the picture doesn't really reflect your biological question, you can work on the data, okay? Just be aware that working on the data does not stress the data or manipulate the data, okay? These are two different things. And this one very important concept to understand, that just because you don't have the results that you expect doesn't mean that the algorithm is wrong first, doesn't mean that what you see is wrong. Maybe it's the only thing that you can see here, or maybe you can do some adjustment. So changing the parameters is not really seen as a manipulation of the data, but you have to be careful, because if you torture a person after 1 year, at some point, the person will tell you whatever you want to hear. Okay, after I filter this thing just for the sake of time so far, and this one is for, I don't know, removing the noise, I will apply a PCA that is like a projection. So from a high dimensional space to a two-dimensional space. I just want to highlight again, this is wrong, okay? It's wrong to use PCA over a single cell RNA-seq matrix, but I just want to show you what is the outcome and why we need other clustering algorithm. Then I apply the k-means clustering algorithm with a k of 5. So I say just find 5 clusters, and then I apply the hierarchical clustering with this method. And then I ask to cut the tree, and actually also here I can say when until you define 5 clusters. So we have 5 clusters here, 5 clusters over here, and then I can plot both of them. So as you can see, since the PCA, the projection is the same, the position of every cell is exactly the same between the two methods. The only thing that changed is the color. And as you can see, the color is kind of different comparing the two clustering algorithm. So yeah, changing clustering algorithm really changed the results. But as I said before, this is wrong. Indeed, if you ever seen a single cell RNA-seq analysis, well, you would have never seen such a plot. I mean, this is an artifact. It is an artifact for, oh yeah, here I plot also hierarchical clustering, the dendrogram, if you want to see a bit more how it divided all of your cells. But why this is wrong? Well, this is wrong for a very peculiar reason. That single cell RNA-seq data are sparse. What does that mean, sparse? That means that they are full of zero. So if you open one table, one matrix count, and you count how many zero there are, well, there are plenty of zero. What are the zero and why they are there? Well, we have a zero. Basically, the main problem is that why do we have this zero? It is a real zero or is a technical zero? Since we have so many genes, I mean, we don't really have the 100% chance to pick all of them. So if here there is zero, I mean, I'm not sure, I can't be sure 100% that this gene in this cell, it is not expressed. Maybe we just sequenced it not deep enough, or maybe the instrument was not sensitive enough to depict this gene. We were just unlucky. I mean, we are talking about an amount of data that still it is really hard to manage and to handle. So having a zero that we are not sure that they are real zero or technical zero in our data, it's really hard. And of course, as you can imagine, a zero can change everything in the statistic. So how you treat it? There are tools that are specifically designed for sparse data. So for single-cell RNA-seq data. That's why you can't use the common tool that we use so far that have seen also for bulk RNA-seq or others, because the input data, they are different for itself. So there are two options. I'm going to show you these two options here, Seurat and SciMPy. One is in R and the other one is in Python. Both of them are valid. As I said before, Seurat is the gold standard, but also SciMPy is very famous. They use a different approach for clustering algorithm, use different parameters. That's just up to you. As always, if you have a single-cell RNA-seq experiment, you have to give it a try. You cannot do things a priori. And as before, you have the QR code again to the GitHub, and let's check the script. So the second one that you will find is Seurat. You will see that most of the time Seurat, it will always it will use exactly the same command line that you have seen from my previous colleague, and this command line is basically creating what is called a Seurat object. So starting from an MTX and TSV file, of course, you could have started already from RDS, because as I said before, RDS is made specifically for Seurat. But since this, I wanted to generalize everything, then I converted everything to this MTX and TSV file. So as you saw before, you can read, in general, a table with read MM, read lines, and read lines for the genes and the barcode. I'm not going again through this. Again, if you are confused about this, these commands, please rewatch the webinar, the previous webinar, so the webinar number 2, sorry. Then here I subsample again, and I pick up 500 cell. And as you can see here, I set the seed. Because setting the seed here, since I asked for 500 random cell, it will be this different if I don't set the seed, if you run it on your computer. But since the seed is set, then this one will be always the same. And then I create the Seurat object. Then I normalize the data, find the variable feature, and scale the data. And these are some command line that you also see last time whenever it comes to normalize data and filtering and all these things. Here I put basically the standard pipeline for Seurat. There are plenty of other options. You can explore them, but as a first approach to do clustering, I believe that this is more than enough. And then, of course, you run the PCA. And this one, it is kind of what we saw before, but it's a bit different. Because this one is actually performed in order to project our data on a lower dimensional space, but it will not be used to visualize the data. Seurat used this object, this function, in order to reduce the space and kind of trying to remove the noise and take only what is important from our data. And then here we can do the cluster through these two function over here that of course use always the Seurat object. And this dimension over here, they are how many dimensions from the PCA we want to use. So whenever we project something from a high dimensional space to a lower dimensional space, that means that we are dividing this object into dimensions. So if you think about the donuts before, the donuts, it is the composition of 3 different axes, Y, X, and Z axis. So when Seurat is doing this run PCA, and in order, is like dividing, secting our data set through dimensions. And then here, we just tell how many dimensions we want to use. So usually there are also ways to visualize this data and see how data are changing according to how many dimensions we want to use. But you can start low with this dimension from 1 to 10, and then you can even modify this value and see how your results are going to change. Of course, as I say, this is a standard pipeline. all these parameters and everything, they cannot be really perfect for every data set. This is the main problem. You have to just give it a try. Such as this resolution. The resolution is another parameter that tells you how the cells has to be close to each other in order to be a cluster, kind of like this. Of course, resolution 0.5 is a good value. It was actually evaluated empirically. You might want to change it in order to have a more global vision or a more local vision of this one. As before, you have to just play with the data and see what's going on between your data. And then you run the UMAP. Now, the UMAP is actually what is used from Seura for the visualization. It's kind of projecting again from a high-dimensional space to a two-dimensional space, but it's not linear as the PCA. It is more precise and it is more, it is what is used so far for single-cell RNA-seq. And then you can plot. So we can see what we have. We have a few warning. You're not happy with the code if you don't have a few warning. That's always like this. Some PC and some genes that are actually important for for the clustering. And after a few things, then you have optimization finished and everything, and then you have the plot. So this plot looks more real about our data. So it's like dividing our cell. There is more. You can see, you can explore this data a bit better. You can check why this cell that it should belong to the cluster 5 actually it belongs to cluster 3. The same for this one that belongs to the cluster 4. We can ask so many questions, and then we can dig into the data. One thing that is really important that I added here in the end is this other code. What this code does is to basically generate a data frame that is a table. It is something like this that tells you this cell belongs to cluster 2, and these are the coordinates. So where this cell belongs to the UMAP. This is important because using only one clustering algorithm is really limiting. So if you want to use, for example, this information for a feature selection or on another algorithm, another program languages, well, you have to export this information. So I added this script that takes this raw object in input, and basically this script, it will write this data frame. So basically what you have here, that it can be taken as input for other algorithm. For example, let's say that we want to change the color of this one. Well, you can load this since you have all the coordinates of each cell. Then you can color them by cluster because you also have the cluster number, but changing the colors through another palette, okay? And this palette, for example, this one is the set 2 that I used. You can use the set 1, a dark 2, and so on, or specify every color for each of them. So that's totally up to you. But you need to extract this one if you want to get creative. This is Seura. So very great tool, super updated every time. And of course, if someone asks me, I also use Seura most of the time. But you also can give it a try to ScumPy. So if you don't find what you expect, well, then you can find ScumPy. So ScumPy is in Python. And as you can see, with Jupyter Lab, I can actually run everything either in Python or in R. Just be aware that whenever you choose a notebook, you have to choose either the Python or the R one. And over here, you have Python 3, for example. Instead here, you have R. So it tells you which is the kernel that is using. For ScumPy, everything is a bit more straightforward. So you basically load the data manually in the program, and then you subsample, and then again, I set the seed as a 42 as before, then I preprocess the data. So I normalized using, all of this is default standard for ScumPy. So whenever it comes to normalization, don't just normalize your data because you want to normalize, but always read what is required from that clustering algorithm. Because every clustering algorithm, either they require the normalization or they don't because they have their internal normalization, or they tells you that you can do whatever you want. Okay, so this is also pretty important in that. Then of course I select the 1000 highly variable genes. So I'm doing exactly the same as I did before, and of course these are log10 transformed into log10 data. And then I scale them, I evaluate the PCA, I evaluate the neighbor and low vein. You don't really need to know in the detail what all of these are doing, okay? Because, I mean, you want to make a clustering, you want to evaluate your clusters. Of course, knowing how everything is working precisely, it will help you from a point of view, but also from another point of view, it will be pretty impossible to know every kind of clustering algorithm or whatever they do. So you can give it a try. This is my best suggestion about from this part. If you're really desperate, then you have to dig into the data and you have to dig into the code. And then, of course, you can save the results of the cluster, that it will be kind of the same file as I showed here. And of course, you can use the UMAP to plot and project your data. And as you can see, I mean, the two pictures are not the same. And there is not really a strict way, a direct way to compare the data, the two clusters, the cluster obtained from this one and the cluster obtained from this one. What you can do, because also the value here, 0, 1, 2, 3, they don't really, actually, so Seurat, for example, has 6 clusters over here. Instead, ScanPy has just 4 clusters. So, I mean, how can you compare them? So the only thing that you can do is to check if every cell are in the same cluster in this one and in this one over here. But I mean, it will be a bit more complicated. So the best shot that you can have is that after you generate the cluster with Seurat or with ScanPy, then you proceed. You proceed to the feature selection. That is what we'll talk about Sophia after me. And proceeding to the feature selection, you will see the genes that are more important for each of these clusters. And then you will realize if Seurat represent better your data, or if ScanPy represent better your data. So, said this, I believe I told you everything from my part. I know it is very general, but I hope I gave you the idea about what is a clustering algorithm, why you have to do clustering, and how to pick up the clustering algorithm in a way. So you know that you have to arrive until the end of the analysis before to say, okay, this is the clustering algorithm that I want to choose. As a last thing, here I showed only 2 tools, Seurat and ScanPy. So this, you can install it through our install packages because it's from CRAN. This one you can install it by pip. Everything is already installed here, and this Docker that you can download, so you don't need to install anything. And for the day of the biohackathon, the machine that you will use, they will already have at least Seurat and ScanPy already installed. My suggestion, though, is that please, before to arrive at the biohackathon, read a couple of other tools to perform a clusterization. Because, of course, for whatever you will do that day, that I cannot make any spoiler, but you will have to get creative. You cannot solve the problem that you will have to solve directly with Seurat or ScanPy. You have to get creative, changing maybe a few parameters or changing the clustering algorithm. There are several clustering algorithms that you can take a look that are really worth the look. One of them is called CSVI, and the CSVI is a tool that uses autoencoders, so the deep learning, in order to project the data from a high dimensional space to a low dimensional space. So just take a look and have fun. And I will, maybe we can take a 5 minutes break, and then I will leave the stage to Sophia. Thank you.
Hi. Okay. Hi, I'm Sofia, and I'm going to talk to you about single-cell RNA-seq computational method, the second part that is related to evaluate your clustering algorithm in terms of characterize cluster biologically. The code that we will see later is available here, and it is a GitHub repo. So, since, as Luca told us, we have this cluster here, so our cell grouped together, and we want to characterize this cluster in order to evaluate if the clustering algorithm is able to capture the biological properties that we are looking for, or simply if we we can compare the different results for different clustering analysis. So mainly what we want to do is to identify the so-called marker genes, that are genes that are able to characterize a cluster compared with all the other clusters. And this is important because thanks to these marker genes, we can link gene expression to specific cell type and specific function. So for example, we want to say if this cluster belongs to epithelium tissue, or it belongs to bones or something else. And this is important because it provides interpretability. The first step, the first analysis that we want to run in order to evaluate our clustering analysis is differential expression analysis. This analysis is able to identify the so-called marker genes, so genes that are really expressed in one cluster if we compare this cluster to the others. Okay, so now a brief introduction, a brief description on how differential expression analysis work. We mainly adopt statistical models in order to rank our genes. So we have as input our normalized gene expression matrix, and also the labels for each cluster. So each cells of our dataset is associated with a specific cluster. And we want to rank genes and characterize each genes with a log two-fold change. So a difference in terms of expression between one cluster and all the others. Then we can associate to this, since it are statistical tests, we can associate this test with a p-value and other justice p-value. Also, it is possible to associate other kind of scores. The common statistical models that are used are Wilcoxon-Rank test, that is a non-parametric test, or many others, but there are a huge set of tests that you can run on your data. I reported here some of the most common used. Each one has some kind of peculiarity. So, for example, there are tests that are dropout aware, that is really important for single-cell data, or tests that are able to take as input directly the row counts matrix. And also there are tests, like this one there, that are able to take into account covariates, so are able to select a set of markers together. Now I want to present this test that is pretty simple. And in my experience, for many applications is fine. So it's super fast to run. And this test here is, as said before, is a non-parametric test. This means that we are not assuming any kind of knowledge about the distribution of our data. So how does it work? Mainly, we have we start from gene X, and we rank all the cells according to their expression value. If the cells that belong to a specific cluster are ranked are mainly occupying the highest rank, this means that this genes is able to separate the expression from one cluster to another. So we can imagine that we can calculate a score based on the rank that each cells reach, and assign a capacity of these specific genes to mark a cluster against all the others. Also, this is a statistical test, so it is possible to measure a p-value, and take only the genes that has a significant p-value, so has a high probability of observing their rank in their highest rank. It's also important to notice that we need adjusted p-value to correct for multiple testing, since we are testing many genes all together, and take under the control the false discovery rate. Okay, now we have this ranked genes list, and we want to what we can do with this ranked genes list. On one side, we can do cell type annotation. On the other side, we can do functional enrichment analysis. First, we start with cell type annotation. That is a computational method that is used to assign some kind of biological identity. So, for example, all those cell type listed here to a cluster. There are two ways in which we can do cell type annotation. The first one is manually created. So you already know some important marker for your data, or you take it from the available resource. Most common are Pangao Lab cell marker, Pangao DB cell marker, human protein atlases. And you just check if there is a marker that is specific for a certain cluster. But this is manually, so we have a huge amount of genes in our dataset, so it's pretty hard to do this kind of analysis. So there are some tools, for example, Singular, that is based on R, and is available on R, and that are automated and simply try to match expression profiles to reference dataset. Okay, let's focus on Singular. We have to run Singular. We just need to focus on one cluster each, or we can focus on all the cluster together. But we take one cluster. For this cluster, we compute some kind of statistics, some kind of value that are representative for the cluster. For example, we compute the centroid, and then we compute the correlation scores, Spearman or Pearson, between already known profiles. So we have a database in which are stored different different cell profiles, so different RNA-seq for cells, and we compare our centroid to this kind of profiles, and then we assign the label of the most similar reference cell type. As you can see here, we have the cluster, and then we have the association assigned by Singular. Okay, let's move to some code for this kind of analysis. This is our code. So mainly, as Luca said before, we have this row here that simply does some preprocessing and then compute one type of clustering analysis. This, as Luca said before, I adopted here Seurat. Now we have the association for our dataset to the different cluster. That is just a label. Now we arrive to this part of the code here, in which we create a single cell experiment object that is another packages inside of R that is able to take, that is useful because thanks to this object here, you can run singular. And we take a reference, so human primary cell atlas data, but you can choose a different kind of reference. Also, if you have data that comes from mice, you can choose mice data set instead of human, and so on. And then we have we run in here the analysis, so singular. And what happens is that to each, this is another human, that each cluster would be associated with a specific cell type based on their centroid. Of course, you can use different kind of matrix to select one cell, one representative cell to each cluster, but for this case, centroid could be fine. Okay, let's go back to the presentation. Okay, let's move now to a different kind of analysis, that is functional enrichment analysis. This one is based on gene ontologies. So the idea is to identify gene ontology terms that are statistically overexpressed in a specific list of genes of our interest. For example, genes that are upregulated in a specific cluster. So thanks to differential expression analysis, we have this ranked gene list. We can set some threshold, so based on log-fold change or based on the number of markers we want to take into account, and select a subset of genes. Now, what we want to evaluate is that if there is an enrichment of specific gene inside our set of genes, set of markers previously selected. So in this plot here, what we have is our input gene list, and then we have another k genes that are the annotated terms, so the genes that are well known to be, for example, biological process, molecular function, are super expressed in some specific molecular component. And we calculated the intersection between our genes list and our input genes list and these geo terms. Now, once we have this intersection, what we want to evaluate, and also we have the universe of all genes that we have at the beginning of our analysis. So, for example, as Luca explained before, the genes that are highly variable inside our dataset. Now, what we want to evaluate is if we try to keep by chance, so randomly, we select randomly exactly k genes from the universe, how many of these genes also belong to the geoterms. So in this case, what we have is that we have some small p-value, so some small probability, indicates that the overlap between your input genes list and the geoterm you are taking into account is greater than the one expected by chance. So this means that we are able to see an enrichment. We can test many of these genes, so the geo terms, independently, and because we test a huge number of geo, it's important to correct for multiple hypothesis tests. What is a gene ontology? Gene ontology is mainly a group, a set of different gene sets that describe so or are annotated, and we know that are super active or super expressed in specific pathway. For example, biological process. For example, molecular function or cellular component. There are also two important resources here that are reactome and KEGG. And those two are mainly based on pathways. So also not only simple gene set, but are direct graph. And Reactome and KEGG are really important because are manually created. So, and also are manually annotated and takes into account a huge variety of information. Okay, let's move now to a second kind of functional enrichment analysis that is called GSEA. For GSEA, it's pretty different because in the first case, we have the ranked gene list. We select a subset of genes and we analyze if this subset of genes is in some way enriched for a certain specific gene set of interest. In this case, for analysis, for GSEA analysis, what we do is that we take into account all the ranked genes list. And what we want to measure is if this gene, in our ranked gene list, the genes that are mainly on the top or on the bottom belongs also to a specific gene set. So as you can see from the plot here, what we want to measure, for example, in this case, is that we have a gene set that is each bar of this plot, and is known to be some specific process. And what we want to measure is if all the genes belong to the top expressed or to the top not expressed genes for our cluster. So we can call this this score here running enrichment score, and we can associate to each gene set we take into account a running enrichment score. It's important also to normalize this enrichment score because each gene set we can take into account has a different length. So to compare the results for different gene sets, it's important to normalize them, the score on the number of genes that they belong. And we have the same settings described before. So also in this case, it's important to correct for multiple hypothesis test. For functional enrichment analysis, GSEA, we have this gene set here that is a molecular CINI database that is a really huge collection. In my experience, the one that I used more are hallmarks and C2. That is inside C2 we also have these two datasets, so Reactome and KEGG. Okay, let's move on the code. So we have this cluster. Okay, let's okay, go on with the plots. Okay, so let's move here. Okay, so we mainly have our data that I called combine because I took two from the whole, from the entire data set at the beginning. I took just some example and I combined them. And then we have our function to find marker that is running with a Wilcoxon rank-sum test. And then we can do some step of pretty easy preprocessing. And now we want to run our geo-analysis. So for example, in this case, we want to take this kind of ontology, this is molecular function, and we want to run the correction. And the plot that we have as output is this one here on the right, in which we have, for example, this is for molecular function. So we have a list of specific molecular function for which some genes, some marker genes are already known. And we can see that we have here the p-adjusted value. And what we can say here is that, for example, for cluster 0, we have an enrichment in structural constituent ribosome or some kind of process related to cell cycle phase or some proliferation step. And in this case, the analysis was quite good because we have some enriched positive and some, we have both positive and more not so good enrichment. And yeah, that's for this kind of analysis, you have to focus on what you already know on your data. So if you expect that there are a specific cluster that is related, for example, to a specific cell cycle phase, and you have to know a priori which genes that you expect to see here. So you expect to see enriched here. Okay. Let's move now to the other kind of gene set enrichment analysis, that is GSEA. The code for GSEA is here. So for this step, you need to, of course, download the gene set, for example, in this case, hallmark, but you can download many other gene sets here. And once you have downloaded this data, you need to select some specific. You need to do some specific step in order to have the gene, to convert the gene name into gene symbol and so on. But you can also take gene name, but it's the same. And then you can run the analysis. In this case, I decided to cut all the p-values that are under a certain threshold, so keep only the significative gene set, the genes that are significantly enriched, and we can run the analysis. Okay, and we have this plot here that is pretty similar to the others, but in this case, the terms on the y axis, the term here are different because are related with hallmark, and as you can see here are pretty different from the other because they are more specific with metabolism, phosphorylation, so specific to process and not to as before with some kind of cellular component. Okay, and also in this case, the plot is pretty the same. So we have this ball here that represent a p-value and the number of genes that we have we found enriched in our dataset. Okay, so on just to show you better the results. Okay. Okay, the code is not running correctly. Okay, I'll go there. Okay, so mainly in this object that is called hallmark, or as you prefer, but in which you store the result for the GC analysis, you have many different columns. In the first one, you have the results, so in which mainly you have an ID for each gene set tested. And for each gene set tested, also you have a description, a set size, so how many genes you found enriched in your dataset, the enrichment score, and the normalized enrichment score, the p-value, and the p-value adjusted. What is also important here is that you have the core enrichment, so a list of genes that you found really expressed in your cluster. So this is important because you can also compare because in some case, maybe you found an enrichment for example, a gene set, for example, this one, both in more than one cluster. So in this case, you can go to the core enrichment and check if the genes are also are the same or are different, and measure it in the power, and measure their power to be marker genes. Okay. Let's go back to our slide. Okay, what I want to say is, okay, let's start, go back here. And what I want to say to conclude this presentation is that why all this analysis that we saw today are important, because if we have because since we have this cluster here, and we want to characterize its cluster, we need a huge, big set of analysis to do this job. So it's not an easy job, but all those kind of analyses are pretty useful to understand what's happening during the cluster. So let's say that does only cell type annotation is not enough in most of the case. And in most of the case, you need more specific analysis. And also you need some kind of, you need some kind of previously experience on the dataset. So some kind of information related with the experiment are why the dataset has been collected and which kind of process you expect to see in the dataset. And okay, so if you have any kind of questions about my presentation and about the presentation of Luca, you can submit them. And we are going to address the question during the last session, so during the session of the 27th of May. And you can submit on the form that you find thanks to this QR code. And thanks for the attention.